Hi, let's study about the second part of the chapter where we'll be learning components of a cell. This part, the learning of the components of a cell will be divided into three parts. Let's dive on to the first part and let's learn about the cell membrane. First of all, before diving into the cell membranes, let's study about the essential components of a cell. Of course, there is the plasma membrane or the cell membrane, which we will be learning in detail in this part. Then there's the cell wall. This is also an outer layering, an outer covering, but while the plasma membrane or the cell membrane, they are present in animal cell, and this is the only single membrane covering the animal cell. The cell wall is an additional membrane that plants have. Okay, so plants have cell membrane or a plasma membrane as well as cell wall. There are two outer covering in plants. And in animal cells, we only have the cell membrane. There is no cell wall in animal cells. We have the cytoplasm. Cytoplasm is that fluid-like component, fluid-like matrix that is present inside the cell membrane. Okay, so and in whatever organelles, whatever entities that cells have, they are embedded inside these inside this cytoplasm. Cell organelles, we have first nucleus, then we have endoplasmic reticulum, which we also call ER, then we have Golgi apparatus or the Golgi complex, okay, Golgi apparatus or the Golgi complex. Then we have lysosomes, we have mitochondria, we have plastids, we have vacuoles, we have ribosomes, and we have centrosomes. In this, plastids is also found in plants, okay. So there is no distinction of what is found in plants and what is found in animals in this slide, but while we are discussing the different organelles, if there is a distinction that this particular organelle is found in plants, I'll be explaining you that, and I'll be explaining you the function as well. For now, let's remember that there is an outer covering in animal cells and there are two outer covering in plant cells, that is the cell membrane plus the cell wall. Then we have the cytoplasm, that is the fluid or the matrix-like structure in which other organelles are embedded or suspended. And then we have 9 organelles over here: nucleus, endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids, vacuoles, ribosomes, and centrosomes. In this first part, where we are discussing the components of a cell, first of all, we will be learning about the cell membrane and the cell wall and the cytoplasm. So let's get started. This is the pictorial representation of the essential components of a cell. You can see here the various components. This is just to give you an idea of how they look like, so that when you visualize, when you have to learn about them, when you have to remember them, there is a visual image. There is a visual image forming in your brain, and that, trust me, that visual image is gonna help you a lot. That visual memory is going to help you even remember some functions which you may think that, oh, I don't remember, but if you try hard enough, you may be able to. Plasma membrane or cell membrane is a biological membrane separating cell components from its outside surrounding or external environment. So basically, they are the outer covering, they are the outer layer that provides protection. From one cell, okay, from this one cell to whatever is present outside this cell. There is this covering. So you may say that this protection, this covering of the cell is like, you may imagine it, you may visualize it like it's home, right? Because home is where we are protected, where we are safe from the outside surrounding. All right. So the home, the place of all these organelles, all right, or these components of the cell, they are all enclosed, they are all covered by this outer layering of cell membrane. This is a biological membrane and it separates the cell contents from the outside environment. Property of the cell membrane is that it is thin walled, it is elastic, it is semi-permeable, and it is made up of proteins, lipids, and it is a living component of the cell. This means that, what do we mean by semi-permeable is that this cell membrane, like if something wants to enter inside this cell, all right, so not all components will be allowed to enter. The property that the cell membrane is semi-permeable in nature, it means that it allows only certain components to enter inside the cell. Alright, it's like when you're at your home and when someone rings your doorbell and you don't want that uninvited person to come inside. Alright, so you may not open the door. Alright, you'll only open the door to those people whom you want to come inside. Similarly, cell membrane over here, it is semi-permeable in nature. That means it allows only selective materials to flow in as well as outside of the cell. This is also important, not only flowing in but also outside of the cell. Not all the organelles that are present over here inside the cell, they may not just go out of the cell. All right, there is some specific molecules, let's just say, or some specific entities only, that during some important processes they are allowed to go outside. So this is owing to the semi-permeable nature of the cell membrane, and it is made up of proteins and lipids. It is living in nature. It functions and it protects the cell contents from its outside environment. Movements of substances in cell membrane. Now that I have told you that the cell or the plasma membrane is semi-permeable in nature and it allows only selected materials to go in and out, let's learn about how this process of going in and out occurs. It occurs via two methods. One is diffusion and the other is osmosis. Diffusion is basically the movement of molecules from high concentration to low concentration, and it is mostly seen in cases of gases. So the gaseous component, when they want to move inside or outside of the cell, it happens from the higher concentration region to the lower concentration region, and it also happens mostly during respiration, because during respiration cells, they inspire, they take in oxygen and they breathe out carbon dioxide. All right. So when this process of breathing in of oxygen and breathing out of carbon dioxide is happening, it is happening through the process of diffusion. Osmosis is the second process, and it is basically the movement of solvent molecules. If you may remember, solvent molecules are those molecules in which the solute is being dissolved. All right. So solvent is always, the quantity of solvent is always greater than that of the solute. All right, so let's just say water, because water is a universal solvent. All right, so if a water is supposed to go inside the cell, or if water is supposed to go outside of the cell, then the process by which that will happen, that process is osmosis. And here as well, osmosis also happens from high concentration to low concentration, as we have already seen in diffusion. How do substances like carbon dioxide and water move in and out of the cell? Discuss. We have just observed this, that carbon dioxide, since it is a gas, so this is going to move in and outside of the cell by the process of diffusion. And how? From high concentration to low concentration. Okay. Water. Since water is a solvent particle, right? It is a solvent molecule. Okay. So the process by which water will move in and out of the cell will be osmosis, and this too will occur from the high concentration to the low concentration. Okay, gaseous particles, diffusion, solvent, or let's just say liquid particles, osmosis. Why is the plasma membrane called a selectively permeable membrane? Because it allows only some selected particles to go inside and outside the cell. All right. So whatever particles that are flowing in and out of the cell, they are basically regulated. That movement is regulated by the plasma membrane, and that is why the plasma membrane is called a selectively or semi-permeable membrane. Let's perform an activity to demonstrate how osmosis happens. For this, we have to take eggs and we have to remove the outer hard covering of the egg. We're gonna do it by dissolving the egg in dilute hydrochloric acid. So the hard part of the cell, that shell, is made up of calcium carbonate. And when you dissolve it in dilute hydrochloric acid, you get calcium chloride. Okay. you get calcium chloride, and calcium chloride, it is easily, it easily removes it will easily remove itself with water. All right, you don't need any other scrubbing or any other peeling in order to remove it. Okay, so we are removing the hard shell by dissolving the egg in dilute hydrochloric acid, and then after that calcium chloride and the shell is removed, then a thin outer skin only would now enclose the egg. That means the egg only contains a cell membrane now. Now we will put the egg in pure water. Pure water here is our solvent particle. So we will put the egg in the pure water, and we will observe what happens after 5 minutes. We observe that this egg is observed to swell up and become bigger. This demonstrates that osmosis has taken place. That means since inside the water there is high concentration of solvent molecules, and inside the egg there is low concentration of solvent molecules. All right, so the cell membrane, owing to its semi-permeable nature, is going to allow the water, the solvent molecule, to move inside the cell, that is from the high concentration to the low concentration. So the eggshell made up of calcium carbonate reacts with hydrochloric acid or HCl to form calcium chloride, a soluble compound which washes away. The cell membrane lying beneath the shell is now permeable to a few things while impermeable to others, and when put in water, osmosis happens, and that is why the egg is observed to swell up. Another experiment to demonstrate how osmosis takes place, they have to place a similar D-shelled egg, that is the egg that has been reacted with dilute hydrochloric acid, in a concentrated salt solution, and we have to observe what happens. Since in a concentrated salt solution, concentrated solution, the solute particle is more than the solvent particles. All right. So the solvent particles is less and the solute particles is more. That is why the solution is concentrated. So there will be more amount of salt in that solution. And since there is less solvent, so what happens is that since osmosis takes place from high concentration to low concentration, so inside the egg there is a higher concentration of solvent than outside the egg. All right. That is why water or the solvent particles will flow from the egg towards outside. All right. So the saline water has more osmotic pressure than inside the egg. As a result, water passes out of the egg solution into the salt solution because the salt solution is more concentrated, and that is why, because the egg is losing water, so instead of being it's in its normal shape, or instead of swelling up, it is observed to shrink. Osmosis with dried raisins or apricots. In this experiment, firstly we will put dried raisins or apricots in a bowl of water, and after that we will observe that raisins are all swollen. All right, they are swollen because water has a higher concentration of solvent, right? So from the water, from its outside surrounding, water is going to move in towards the raisins, towards the apricots, towards the dried material, and they will swell up because of the process of osmosis. All right. And after that, when you place them into a concentrated solution of sugar or salt, in any solution whether in which the concentration of solvent is less, then what will happen? Those swollen raisins, they are gonna shrink because they will lose water. The water will flow from that high concentration, that is from inside the raisins, towards outside where the concentration of the solute is higher. So that is what happens, that when placed in concentrated solution, it loses water and it consequently shrinks. Now let's talk about cell wall. Cell wall, as I had told you earlier, cell wall is found in plants. It is not found in animals. It is a structural layer just outside the plasma layer. So if this is your plant cell, there will be cell membrane, and then there will be another membrane, that is the cell wall. Okay, so cell wall is flexible, tough or rigid in some cells. Non-living, they are non-living. So while the cell membrane was living in nature, cell wall is non-living in nature. It is made up of cellulose. Cellulose is a hard material and it is freely permeable. That means it easily allows particles to go in as well as go out. functions? It protects the cell membrane. So if this is the cell membrane, okay, the outer covering is the cell wall. This is non-living and this is living. Responsible for maintaining the shape and size of the cell, prevents the cell from becoming flaccid and drying out. Another important, since it is made up of a hard material like cellulose, so cell wall, it not only protects the cell membrane, but it also protects the cell from being flaccid, being shrunken, and drying out. So it helps the cell to maintain its integrity, its osmotic integrity. For this experiment, we have to mount the peel of a raw leaf in water on a slide, and you have to examine the cells under high power of a microscope. And you have to notice those small granules which we have called here chloroplasts, since they contain the green pigment chlorophyll, and you know that chlorophyll is responsible for the process of photosynthesis in plants. Then you put a strong solution of sugar or salt on the mounted leaf, and then you have to wait and observe. So basically you are putting that leaf into a concentrated solution, right? Strong solution means concentrated solution, that means solvent molecules is less. So what do you expect? Before that, let's look at another setup, that after you have done that, you place some raw leaves in boiling water for a few minutes, okay? Because you're boiling, that means naturally you're killing the cells. Then mount one leaf on a slide under the microscope, and then you observe. Then you observe while putting that into the concentrated solution, and then you observe whether the osmosis, or which we also call the plasmolysis, whether this osmosis or plasmolysis is occurring or not. In the first case, cell membrane is intact. This is a normal cell, all right? This is your normal leaf cell, okay? Leaf cell which you have before you are doing either of the two experiments. So when you put that cell in a salt or a sugar solution, you have seen that the cell membrane has shrunken because it has lost some water. And when you have again put it in boiling water, so although it has now been put in a place where there is high concentration of solvent molecules, but because the water is boiling water, it has naturally killed the cells. So because once everything is deadened now, so the process of osmosis is not occurring. That means water is not going inside the cell. That is why there is no absorption of water after boiling of the leaf. So what do we observe? We observe that only living cells and not dead cells are able to absorb water by osmosis, and that this occurs because the cell is alive as long as its protoplasm is functioning. Once it is deadened, the process of osmosis, diffusion, any process will not happen. A living cell, it responds to the outside condition, a deadened cell does not. Cell walls, it permits the cells of plants, fungi and bacteria to withstand very dilute hypotonic external media without bursting. In such media, the cells tend to take up water by osmosis. The cell swells, building up pressure against the cell wall, but the cell wall also exerts an equal pressure. So what it is trying to say here is that we know that plants have cell wall, okay? Bacteria, since some bacteria are unicellular in nature, so owing to their one cell existence, they have an extra layer of protection. They also have the cell wall, and fungi also have the cell wall, since fungi also needs that extra protection to survive in the outside surrounding. So when you put all of these plants, or be it fungi or be it bacteria, in a very dilute means where there is a lot of solvent molecules present. So of course osmosis is gonna happen, but so any cell would absorb water to a limit, right? After it has say swelled up to say double, triple its size, there will come a point where it will no longer be able to take up any water and it's gonna burst. But the cell wall, what function it owes is that it prevents this bursting up of the cell. That means when the osmotic pressure inside the cell has been built, that means when the the cell already has a lot of pressure, when the cell is already exerting a lot of pressure inside itself because of the higher amount of solvent molecules now that it has absorbed. But the cell wall, it is gonna exert that equal amount of pressure inside the cell. So the osmosis, because of the osmosis, the solvent molecules are trying to exert pressure outside, but the cell wall is gonna exert an equal pressure inside. So that pressure is getting balanced. That is why this cell wall is able to protect the cell from bursting. Okay. Now we are gonna come to the second part of the chapter where we will be discussing the components of a cell. We have discussed till now the cell membrane, the cell wall. Now let's learn about what a cytoplasm is, and let's learn about the different cell organelles in this second part of the chapter. Cytoplasm is a thick, semi-solid like jelly-like substance enclosed by the cell membrane containing all the cell organelles except for the nucleus. Okay. So in the cytoplasm, that fluid or matrix or even solid jelly-like structure, all the cell organelles, they are embedded. They are they exist inside the cytoplasm, and all the cell components, they except for the nucleus, all the cell components that are enclosed inside of the cell membrane, okay, they are present in that cytoplasm. All right. Property is that the cytoplasm mainly substitutes salts, proteins, water. This aqueous liquid, salt, protein, water is called cytosol, and this facilitates protein synthesis and other metabolic reactions in it. So whatever reactions, metabolic reactions are going to occur inside of the cell, they are going to occur in this cytoplasm, and whatever synthesis is going to happen, mainly protein synthesis, everything is going to occur inside this cytoplasm. So this facilitates that site. Fill in the gaps in the following table, illustrating differences between prokaryotic and eukaryotic cells. So the size, we know that prokaryotic cell is generally small. It is 1 micrometer small. Micrometer small means it is usually, this is 10 to the power minus 6. So it is usually 1 into 10 to the power minus 6 meter. This is, how small a prokaryotic cell is. Whereas eukaryotic cell is generally larger. It is generally 5 to 100 micrometer. Okay. Prokaryotic cell, the nuclear region is dash and known as dash. Eukaryotic cell, the nuclear region is well defined and is surrounded by a nuclear membrane. So we know that there is no membrane bound organelles inside the prokaryotic cell. So and we also know that there is no nucleus or nucleolus inside the prokaryotic cell. So the nuclear region in the prokaryotic cell is not well defined. We may say that, right? And we may say that this undefined nuclear region we call in the prokaryotic cell to be nucleoid. Okay. Chromosome, we have single chromosome in prokaryotic cell, whereas there is more than one chromosome in the eukaryotic cell. In the prokaryotic cell, membrane-bound cell organelles are absent, and whereas in the eukaryotic cell, membrane-bound cell organelles. That by this we mean that organelles, entities inside the cell, they also have membrane. Okay, so membrane-bound cell organelles, they are present in eukaryotic cell, whereas they are absent in the prokaryotic cell. So there is an outer covering of cell membrane as well. The inside membrane, the inside organelles, they also have membranes of their own. Not all of them, but yes, some they do have. But in prokaryotic cell, there is no membrane-bound cell organelle. Let's talk about the nucleus now. Nucleus is an organelle that is found in eukaryotic cell and contains the genetic material of a cell. It is spherical in shape. It is enclosed in a nuclear membrane. It contains nucleoplasm. It contains chromosomes, and it regulates so much. Owing to this, if you may remember, in one of the earlier slides, I had told you to visualize the nucleus like to be the brain of the cell, right? Why? Because inside the brain, the main things, thinking and the main important fundamental process that they happen, right? So inside the nucleus of the cell, the important functions are happening. What are these important functions? That they are regulating cell cycle. What do we mean by cell cycle? We mean the division, we mean the growth, we mean the reproduction. And reproduction, growth, division, all of these, this is important. And without the multiplicity of cell, say suppose if you're injured, so what happens? Those cells, those cells, they are injured, they die, and there is ultimately new cells coming up. So after a few weeks, you may find that your skin is normal again, and there is no blood coming out, or there is no other scratches on your skin. Why is this happening? This is happening because of reproduction of those cells, because of the division of those cells, because there is some growth in these cells, there is some division in these cells. And nucleus is that organelle inside the cell that regulates this cell cycle. So you may imagine how important this is. So it's responsible for cell cycle, it's responsible for cell division, it's responsible for protein synthesis. Protein synthesis, our DNA, like if you may remember that I had told you that chromosomes contain those hereditary particles called genes, and genes, they have DNA, and the DNA, they ultimately participate in the process of protein synthesis. So all of these, they are present inside the chromosomes, and these chromosomes, they are also present inside the nucleus. Okay, so everything basically is originating from the nucleus. So you have growth, you have metabolic activity of cell components. Yes, this is important, all the activities of the cell components itself. Now we know that cell is basically regulating our whole existence, right? But there is one entity that regulates this, the metabolic activities at the very microscopic level, at the very cellular level. And nucleus could be that head, you may say, that that head-like figure that is, regulating, that is basically supervising all of these activities. It stores genetic information of an organism in the form of chromosomes. Okay, this I had explained you in pretty much detail in one of the earlier slides. So it stores hereditary materials in the form of DNA, which is present in genes, and genes in itself is present in the chromosomes. So that is why I had told you to visualize, because if you visualize the nucleus like the brain, you may be instantly reminded, oh yes, that this nucleus is sort of like a head figure of the cell, and it's going to perform some very important functions. All right. Nucleus has a cytoplasm of its own, and that cytoplasm is called nucleoplasm. All right. I am using the earlier word cytoplasm to describe it, so that you have that idea that you have that semi-solid like jelly like substance in which other organelles are embedded. Right, let us take a glass slide and a drop of water on it. Then you, using an ice cream scoop, gently you scrape the inside surface of your cheek like this. All right. Does any material get stuck on the spoon? Now, if you like take a spoon and you try to scrape the inside surface of your cheek, then immediately, of course, cells are not visible to a naked eye. All right. So you may think that, oh, nothing is there. But when you transfer this material, to a glass slide, and that is to be placed under a microscope, and before that you color it with a drop of a methylene blue solution. Again, we are coloring it so that those translucent, transparent, cells are visible to us. Okay. Now the material is ready for observation under microscope. Do not forget to put a cover slip on it. And what do we observe? We observe a darkly colored spherical or oval dot-like structure near the center of the cell. This structure observed is called the nucleus. This dot-like structure is called the nucleus. Under normal conditions, it appears as a double membrane structure with thick dense material inside chromatin. Chromatin, your chromosome. Okay, and when cell division is going on, when say cell division, reproduction, growth, etc., this is going on, since chromosomes have those genetic codes that divide itself. All right. So when that is happening, that is naturally going to be more prominent. More prominent means more visible. So when cell division is happening, nucleus appears bigger with different chromosomes visible. Okay. Endoplasmic reticulum is a network of membranous tubules, like you can see here in this diagram, within the cytoplasm of a eukaryotic cell. So endoplasmic reticulum is not to be found in the prokaryotic cell, it is to be found in a eukaryotic cell, and it is continuous with the nuclear membrane to the plasma membrane. It usually has ribosomes attached. So what happens is that if you see this diagram over here, the nucleus, this is the nucleus, okay. So the endoplasmic reticulum, it attaches itself towards the nucleus on one side, and towards the other side, towards here in the end, it is attached to the cell membrane. So it is continuous with the nuclear membrane to plasma membrane, and it usually has ribosomes attached. So all those endoplasmic reticulum that have ribosomes attached, they are called rough endoplasmic reticulum, and all those endoplasmic reticulum which do not have ribosomes attached, they are called smooth endoplasmic reticulum. What are ribosomes? We will be learning about them later, don't worry. Property, it consists of smooth tubular structure like this over here, and the nuclear envelope. ER gives internal support to the cell. It enables the synthesis of proteins, lipids, and detoxifies any drugs or toxins. So at first you may think that there is a lot of functions to remember, but slowly when you try to divide each component, when you try to visualize each component, where its location is there in the cell, how does it look like, you will slowly and gradually, those functions are gonna themselves come to your mind. So please remember the importance of visualization. Golgi apparatus, it looks somewhat like this. It has, tubular hollow structure known as lumen, and they have this when lumens, they are stacked horizontally to each other, this sort of structure, this appears, this is your cisternae, and then there are other individual smaller spherical bodies that are scattered here and there just outside of this cisternae. These are vesicles. So all this incorporates the Golgi apparatus. Golgi apparatus or Golgi body is a complex of folded membranes and vesicles found in the cytoplasm of eukaryotic cells. Remember this. It is composed of flat sacs known as cisternae, stacked in a bent semicircular shape. As you can see here, this is stacked in a semicircular shape. It may not be semicircular exactly, but if you are to observe this arch over here, this sort of looks like a semicircular shape. So, and each stacked grouping has a membrane that separates its inside from the cell cytoplasm. So, this is a membrane bound organelle, okay, membrane bound. It modifies, sorts and dispatches proteins for synthesis. It also facilitates the formation of lysosomes and the transport of lipids around the cells. These are the functions, important functions of Golgi apparatus. Then you have the lysosomes. So lysosomes, they are known as the suicide bags. I'll be telling you why. Also over here, lysosomes, they contain important digestive enzymes. So what happens is that during digestion, some food particles, they come towards these cells because they want the lysosomes to secrete these enzymes so that the food can be digested. So what happens, you can see that this is the plasma membrane, and lysosomes, these spherical sacs, they're leaving. Since the plasma membrane is semi-permeable, so it is leaving, and this is the food vacuole inside the lysosome. This is from the cells of the digestive system. These food particles, these are say the food particles, they are coming towards the lysosomes, and they are, you can see here, a transfer is occurring over here. Alright, so inside, digestion is taking place with the help of digestive enzymes. So these digestive enzymes, they help in the breaking up of food so that the important nutrients can be absorbed by the cells in its simplest state. So lysosomes, they are membrane bound organelles found mostly in eukaryotic cells of membranes. These are spherical sac-like structures that contain digestive enzymes, like I had here, like this is the spherical, like this is the sac-like structure. They have the digestive enzymes that aid in digestion, and they are responsible for breaking down worn-out parts of the cell and destroying any foreign substance, be it bacteria or be it viruses. That is why they are known as the suicide bag. So what happens if, although the cell membrane is semi-permeable, But if by chance any bacteria or viruses, they were to come inside, they were to intrude inside the cell and cause harm to the cell components, these lysosomes, they have this important function, they have this important capability of breaking down these parts. But while they are breaking down these foreign particles, they themselves are also getting destroyed in the process. So they're basically sacrificing themselves for the safety of the cell components. Because they are themselves getting perished, therefore they are also called as suicide bags. So I hope that till now we have learned about the cytoplasm, we have learned about the nucleus, we have learned about the endoplasmic reticulum, we have learned about the Golgi apparatus, and now we have learned about the lysosomes. So I hope that these cell components are clear to you till now. We'll be learning all the other cell components, the remaining of them, in the further parts.